Hello. As usual, I am driving home from work. So last night, as a bedtime story for myself, I read the randomized control trial for this new neuromodulation device called the ProlivRX. That's PROLIVRX. And it is a external nerve stimulator device. And it is now FDA approved for depression, major depressive disorder for folks that are already on and have not benefited from their existing antidepressant medications. So this is on the heels of the Flow Neuroscience FL100 transcranial direct current stimulation or tDCS device that was also recently approved for depression. So interesting that we're getting a couple of new neuromodulation devices, and both of them are home-based treatments. So the randomized control trial was decently sized. I think there were around roughly 100 patients in each group, if I remember correctly. And this was a blinded study, sham-controlled, multi-site trial. It was an industry-sponsored trial. It was sponsored by the device manufacturer, which is by a company called Neuroleaf, which is an Israeli company. So they came out with this device, and it stimulates the trigeminal nerve branches along the forehead, as well as the occipital nerve branches in the back of the head. There's actually another device. I'm blanking on the name of it, but it is a trigeminal nerve stimulator device that was FDA cleared for ADHD a while back. And I'll just insert that there was a recent randomized control trial on using trigeminal nerve stimulation for ADHD in children and adolescents that was a negative study. They did not find that the device was beneficial over placebo. But in any case, they point to some studies that by stimulating these nerves, you get some effects on some of the brain circuits involved in depression, in mood regulation, attention networks. You know, at some point, all of the brain is connected. So if you start poking at one end, it's going to have an effect somewhere else. And so I'm not super impressed by that necessarily, but you know, they've got their reasons for why they wanted to test this, and they thought that combining both trigeminal nerve stimulation and occipital nerve stimulation would be synergistic. So the trial, they used pretty high currents compared to tDCS. So tDCS uses 1 to 2 milliamp currents to stimulate through the brain. And in this trial, but it was somewhere in the like 6 to 18 milliamp range. So quite a bit higher, definitely noticeable if you're wearing this device. However, the electrodes are spaced much closer together, and the current is just passing over the superficial tissue. It's not going through the skull like in tDCS. And they're using basically an alternating current. It's a biphasic type of current in contrast to tDCS. So all the electrical properties are a bit different. The sham was basically where they stimulated at a lower current amplitude and a much lower frequency. And according to their blind integrity testing, for the most part, the blind stayed intact. I think about 14% in the active group were able to correctly guess that they were in the active group, and 6% in the sham group were able to correctly guess that they were in the sham group, if I recall correctly. And they did some modeling to show that even taking into account the little bit of unblinding that there was, that didn't alter the main effect, the main outcome. And the main outcome was that they saw a significant effect. The effect they saw was about a 8-point decrease on the Hamilton Depression Rating Scale compared to about a 6-point decrease on the sham arm. So overall, they saw a 2.6, I believe, difference between the sham and the active treatment group. in terms of points decreased on the Hamilton Depression Rating Scale. This translated to an effect size of around 0.7 to zero point 0.8, which is surprisingly high for such a small decrease on the Hamilton Depression Rating Scale. But the effect size crucially depends on the amount of variability that is in the data. And I think the way they designed this trial, I'll probably have another episode on this specifically, was designed to really limit the amount of variability that they have. So the denominator and the effect size would be smaller, and therefore you could get a bigger effect size. So the effect size makes it seem pretty big in comparison to other treatments. So for example, typical SSRIs for depression are around 0.3 to 0.4. So getting an effect size of 0.8, 0.7 to zero point 0.8 is surprisingly large, especially considering that these are patients who are already taking an antidepressant and may have failed other antidepressants. So I can't remember if all of them are technically treatment resistant or if only a portion of them are, but at least some of them are considered treatment resistant by the definition of having failed at least 2 antidepressant trials. So I'm a little suspicious about how they designed the trial to potentially magnify the effect size. If you look at the remission rates, they're much more modest. I think there was like a 20 to 30% remission rate and maybe a little bit more response rate. But nonetheless, if that's really the case, if you're 20% response rate for a pretty benign treatment that you can do at home, maybe that's worth it if that holds up. They did transition the RCT, which I think was 8 weeks long. So their final outcome was measured at 8 weeks. But then they continued in an open label fashion. So after the RCT portion for 8 weeks, when they got their primary outcome data, then everybody was unblinded and they continued the study for, I think it was 16 weeks. And they saw incremental improvement over that additional 16 weeks, which is a good sign.
And I forgot to mention that the protocol was that the patients would use the device for 40 minutes at a time, twice a day, for 5 to 7 days per week.
There were basically no major device-related adverse reactions or effects. So what do we make of this? I've already voiced some skepticism. I think it's clear that pretty much all of our psychiatric treatments do very non-specific things, whether it's ECT or medications or neuromodulation. They're doing very non-specific things. That's why most psychiatric treatments have multiple indications across disorders that most likely have very different brain mechanisms involved. Although, of course, everything is interacting with everything when it comes to the brain. This is an industry-sponsored trial. Although they offer some putative mechanisms for how the device works, I think mechanistically it's just as unclear as how anything else in psychiatry works. I think in terms of mechanism, the things that we have the best sort of idea and are more hypothesis-driven, model-driven is with TMS, where we're able to do functional imaging and also test our models and hypotheses by fairly focal stimulation. The primary outcome wasn't reached until week 8, and then you saw incremental gains after that. So although this might have decent effects, it's not a very great acute treatment for severe acute depression. So things like ketamine or ECT or even rapidly titrated medications would likely be the better choice for a more severe and urgent depression. But if you have some well-resourced patients who can afford such a device, it's worth a try, I suppose. If I'm comparing this device versus the tDCS device, I would lean more toward the tDCS device just because there's been a lot more research on tDCS compared to this combined external nerve stimulator device. And mechanistically, there's a little bit more confidence around how tDCS is working in terms of the brain circuits that it's targeting. But again, it's a fairly harmless treatment. the worst thing that can happen is you waste time and money. So I think for low urgency depression, definitely worth considering. And of course, it's nice to have more tools in the toolkit. All right. Well, hopefully that was helpful. Feel free to leave a comment or question at my blog, brandonbrownmd.com, or you can send an email to me at brandon@brandonbrownmd.com.